Welcome back to Unit 4 of Acing AP Biology. Today, we will be continuing our discussion of organelles in the cell and cell-to-cell junctions. Lysosomes are only found in animal cells. They are involved with digesting macromolecules and recycling the cell's organic material. They also get rid of cellular waste via programmed cell death. Lysosomes contain hydrolytic enzymes which help the lysosomes break down organic molecules. Hydrolytic enzymes function well in a pH of 5, so that is the internal pH of the lysosome. This is why it is so important for each organelle to have its own membrane, because it separates its environment from the rest of the cell. So, the low pH environment in the lysosome won't affect the optimal pH for the rest of the cell. Next, let's talk about the central vacuole. The central vacuole is unique to plant cells. It is surrounded by a membrane called the chloroplast. The central vacuole has several functions. It stores water, exerts pressure as water comes into the cell. It stores organic compounds as well as inorganic compounds like ions. It holds harmful metabolic products, protecting the rest of the cell. It can store pigments and sometimes contain toxins that protect the plant from predators. The peroxisome functions in detoxifying poisons like alcohol and other toxins. It has a single membrane and thus is not part of the endomembrane system. It has a crystalline core where it holds a variety of enzymes specialized for breaking down these toxins. For instance, peroxisomes break down hydrogen peroxide into water and oxygen. The enzyme catalyzes this process. The mitochondria is known as the powerhouse of the cell. It is the site of cellular respiration, a process that converts glucose into ATP, energy for cellular processes. Mitochondria contain their own DNA and ribosomes, allowing them to be semi-autonomous, meaning that they can survive and reproduce in the cell independently. It has a double membrane, an outer and inner mitochondrial membrane. The inner membrane is convoluted, increasing surface area. The folds of the inner mitochondrial membrane are called cristae. The mitochondrial matrix is the space within the inner mitochondrial membrane. The space between the two membranes is called the inner membrane space. The morphology of mitochondria is vital to its function in the cell, which we will discuss in a later unit. Chloroplasts are only found in plant cells. Their main function is photosynthesis, converting light energy to chemical energy for the plant. They are also semi-autonomous because they carry their own DNA and ribosomes. They have an outer and inner membrane, and the space between the two membranes is called the inner membrane space. Inside the chloroplast, the fluid surrounding the thylakoids is called the stroma. Thylakoids are sac-like structures that are heavily involved with photosynthesis. The space between thylakoids is called the thylakoid space. Stacks of thylakoids are called granum. The cytoskeleton is composed of a system of fibers which provides support and structure as well as allow for movement. There are three types of fibers making up the cytoskeleton: microtubules, microfilaments, and intermediate filaments. Microtubules are the largest in diameter. They are hollow and are made up of the protein tubulin. They increase in size by adding tubulin to the ends. They can be built and taken apart anywhere in the cell. Their function is providing support and cellular structure. They also provide tracts for vesicles to move from one organelle to another in the cell. They are also involved with the separation of chromosomes in mitosis and meiosis, which we will discuss in detail in a later unit. The centrosome is the center for microtubule assembly, which exists in both animals and plant cells. Centrioles, however, only exist in the animal cell and are involved with cell division. Microfilaments have the thinnest diameter. They are made up of the protein actin. They provide cell support and are involved in muscle contraction. They also function in the formation of a cleavage furrow during cell division. Intermediate filaments are medium in diameter. They are the most stable out of the three fibers. They are more permanent than microtubules and microfilaments. They are made up of the protein keratin. They reinforce cell shape and anchor organelles, such as the nucleus. They make up the nuclear lamina, which is the nuclear skeleton. Now let's dive a bit deeper into centrioles. Centrioles, as I have mentioned before, are unique to animal cells. They are involved with the formation of the spindle apparatus during mitosis and meiosis, which we will discuss more in a later unit. Centrioles are composed of 9 triplets of microtubules, arranged in a ring with no center. Centrioles can transform into basal bodies, which anchor flagella and cilia. Flagella and cilia impart locomotion in the cell. They have a 9+2 microtubule pattern, meaning that they have 9 pairs of microtubules arranged in a circle and a pair in the center. Now let's talk about the plant cell wall. The plant cell wall is composed of strong cellulose microfibrils. It provides support by exerting turgor pressure. As water enters the cell, it goes into the central vacuole, creating pressure. The cell wall exerts an equally strong force in the opposite direction. This force provides support for the cell. The cell wall also protects the cell. Young plant cells only have a primary cell wall, which is thin and flexible, allowing for further growth and development. After the plant has finished growing, a secondary cell wall forms to fortify the plant. The wall closest to the plasma membrane is the one that is most recently formed. The middle lamella is a sticky polysaccharide that holds plant cells together. It is composed of pectin. We will discuss the role of plasmodesmata on the next slide. We will end this video talking about cell junctions. Cell junctions allow for cell-to-cell communication and the transfer of materials between neighboring cells. Plasmodesmata in plant cells permit the transfer of water, small solutes, protein, and sometimes RNA molecules between neighboring plant cells. In animal cells, there are 3 types of cell junctions: tight junctions, desmosomes, and gap junctions. Tight junctions inhibit movement of particles between cells. Epithelial cells in the skin form tight junctions, as it is vital for skin cells to be sealed tight with no leakage. Desmosomes join cells together in the formation of strong sheets. However, particles are still allowed to move freely in the spaces between cells. And gap junctions are like the plasmodesmata of animal cells. They allow for the transfer of material between neighboring cells, mostly small solutes, not anything too large like macromolecules. That's a wrap for Unit 4. In the next unit, we will go over the plasma membrane, structure and function, as well as transport and cell signaling. See you then. For more bio content, hit subscribe.